Observation of E-cadherin adherens junction dynamics with metal-induced energy transfer imaging and spectroscopy. Dao Chen, Narain Karedla and Jorg Enderlein. In Communications Biology. Abstract. Epithelial cadherin, ECAD, mediated cell-cell junctions play a crucial role in the establishment and maintenance of tissues and organs. In this study, we employed metal-induced energy transfer imaging and spectroscopy to investigate variations in intermembrane distance during adhesion between two model membranes adorned with ECAD. By correlating the measured intermembrane distances with the distinct ECAD junction states, we probed the dynamic behavior and diversity of ECAD junctions across different binding pathways. Our observations led to the identification of a transient intermediate state referred to as the X-dimeric state and enabled a detailed analysis of its kinetics. We discovered that the formation of the X-dimer leads to significant membrane displacement, subsequently impacting the formation of other X-dimers. These direct experimental insights into the subtle dynamics of ECAD-modified membranes and the resultant changes in intermembrane distance provide perspectives on the assembly of ECAD junctions between cells. This knowledge enhances our comprehension of tissue and organ development and may serve as a foundation for the development of innovative therapeutic strategies for diseases linked to cell-cell adhesion abnormalities. Introduction. Epithelial cadherin, ECAD, is a prominent member of the classical cadherin family, categorized as a type 1 cadherin. It serves a crucial role in facilitating calcium-dependent adhesion among epithelial cells, thereby contributing to the formation of adherens junctions on the cell surface 1, 2, 3, 4. By mediating the adhesion between epithelial cells, ECAD contributes to the establishment and maintenance of cellular interactions within tissues. The formation of adherens junctions, enabled by ECAD's activity, promotes the cohesion and structural stability necessary for the proper functioning of organs and tissues. However, the regulation of ECAD expression and activity is not static but rather subject to dynamic changes. These alterations play a vital role in various physiological and pathological processes. 5. For instance, the loss or downregulation of ECAD expression is often observed in metastatic cancers, where it contributes to the acquisition of invasive and migratory properties by cancer cells. 6. In such cases, ECAD's normal adhesive function is disrupted, allowing cancer cells to detach from their original location and invade surrounding tissues. Additionally, during inflammatory responses, the dysregulation of adherens junctions and ECAD expression can compromise the integrity of epithelial barriers. This disruption can result in increased permeability and allow the infiltration of harmful substances, exacerbating the inflammatory process. Thus, the dynamic regulation of ECAD in these scenarios becomes crucial for maintaining tissue homeostasis and preventing the progression of inflammatory disorders. 7, 8, 9, 10, 11. The classic form of ECAD is comprised of 5 structurally similar extracellular cadherin-like domains, EC domains, arranged in tandem. These domains are connected by conserved calcium-binding linker regions. ECAD also possesses a single transmembrane segment and a highly conserved cytoplasmic tail. Numerous studies have underscored the pivotal role of the EC domains of cadherin in regulating the assembly of cell-cell junctions 12, 13, 14, 15. The process of cell-cell adhesion involves multiple steps, encompassing the dynamic transition between various molecular states, ranging from free monomers to transdimers, clusters of ECAD molecules, and interactions with intracellular domains. Fig. 1a, 16. A weak and transient X-dimer has been reported to exist during the adhesion process, acting as a kinetic intermediate that lowers the energy barrier during the formation of strand-swapped S-dimers in adherens junctions 12. Initially, this X-dimer was identified through site-directed mutations in ECAD 17, 18. More recently, the X-dimer was finally observed in wild-type cadherins using cryo-electron microscopy 19. ECADs have been the subject of extensive investigation utilizing various experimental and theoretical approaches, which have significantly contributed to our understanding of ECAD-mediated intermembrane adhesion 12, 13, 14, 16, 20, 21, 22, 23, 24, 25, 26. For example, NMR relaxation dispersion spectroscopy has enabled the measurement of kinetic parameters in the transdimerization of ECAD-EC fragments in solution 12. Single-molecule Forster resonance energy transfer studies have provided evidence of the mutual cooperativity of cis-trans interactions 13-21. Fluorescence microscopy has revealed the involvement of a nucleation process in junction formation 23-27, while reflection interference contrast microscopy and computational simulations have shed light on the role of membrane fluctuations in mediating the interaction between ECAD bonds 22. However, limited attention has been given to variations of membrane-membrane distance during adhesion. Crystal lattice studies have identified EC5 distances of 29 nanometers for the X dimer, 37 nanometers for the S dimer, and 19 nanometers for the cis cluster of ECAD 19, 28, 29. These variations in EC5 distances during adhesion would lead to a corresponding change in membrane-membrane distance, offering an alternative means of monitoring ECAD adhesion. However, resolving such minute distance changes requires a high-resolution technique. Metal-induced energy transfer, MET, imaging and spectroscopy represents a cutting-edge super-resolution fluorescence technique ideally suited for such inquiries 30, 31, 32. MET allows for localizing fluorophores in the axial dimension with nanometer scale accuracy, employing conventional fluorescence lifetime imaging microscopy, FLIM. This technique capitalizes on the electrodynamic near-field coupling between an excited fluorophore and surface plasmons within a planar metal film. The distance-dependent energy transfer from the fluorophore, donor, to the metal, acceptor, in MET leads to a distinctive modulation of both fluorescence lifetime and brightness as a function of the fluorophore's proximity to the metal surface. By measuring the fluorescence lifetimes and employing a suitable theoretical model, we can accurately determine the distance between the fluorophore and the metal. We used MET to dissect the intermembrane adhesion process mediated by ECADs. Our focus lies in exploring the variations in membrane height, particularly the membrane-membrane distance, during the adhesion of two ECAD-modified membranes. By employing this approach, we can unravel the dynamic landscape of ECAD binding states and provide information regarding the behavior and interactions of ECADs during cell-cell adhesion. Results.
We used a biomimetic system to mimic ECAD-mediated cell adhesion, observing adhesion events between partially fluid-supported lipid bilayers, SLBs, and giant unilamellar vesicles, GUVs. The SLBs were prepared using 1,2-dipalmitoyl SN-GLYCERO3-phosphocholine, DPPC, doped with 1 mol percent acyl chain labeled 1-acyl-2, 12, 7-nitro-2-to-1,3-BENZOXADIAZOL 4-YL amino dodecanoil, SN-GLYCERO3-phosphocholine, NBDPC, 5 mol percent 1,2-dialyoyl SN-GLYCERO3, N5-amino-1 carboxypentyl iminodiacetic acid succinyl nickel salt, NTA dogs, and 1,2-dialyoyl SN-GLYCERO3-phosphethanolamine N-methoxy polyethylene glycol -2000 dope PEG 2000 by a Langmuir-Blodgett-Langmuir-Schaefer, LBLS method. Note that the bottom leaflet of the SLB comprises only DPPC to ensure the flatness of the SLB on the MET substrate. The GUVs were prepared using 1-steroyl 2-ol-OYLSN, GLYCERO3, phosphocholine doped with 5 mol percent NTA dogs, 1 mol percent dope PEG 2000, and 1 mol percent ATO 655 head group labeled 1,2-dipalmitoyl SN-GLYCERO3 phosphoethanolamine, ATO 655 DPPE. These membranes were decorated with ECAD extracellular, EC, domains Fig 1b. Control measurements confirmed the homogeneity of the SLBs and showed no evidence of phase separation in GUVs containing fluorescent lipid and NTA dogs molecules. Supplementary Fig 1 and Supplementary Fig 12. It should be noted that while the biomimetic system lacks the complexity of biological membranes and does not fully replicate the intricate behavior seen in cellular contexts, it enables us to observe ECAD interactions without the confounding influences of other cellular proteins, contextual factors, or active processes. ECAD receptors were in the form of a dimeric chimera, with all 5 ECAD-EC domains genetically fused to an immunoglobulin Fc fragment exhibiting a hexahistidine tag, His6. The SLBs were prepared on a substrate consisting of a 10 nanometer gold film and a 10 nanometer silica spacer, denoted as MIET substrate. The distal leaflet of the SLB was doped with the N-nitrilotriacetic acid NTA chelating lipid, which can further bind to the His6 tags of the ECAD. The GUVs were also functionalized with ECAD-EC at a concentration of 5 mol%. To induce membrane fluctuations and increase the excess area in the vesicles, we created an osmolarity difference between the inside of the GUVs, 230 mosm L sucrose solution, and the outer buffer, 300 mosm L phosphate buffered saline PBS mixed with 0.75 m calcium chloride solution. No adhesion was observed in the absence of an osmolarity difference. Supplementary Fig. 3. The high dye concentration, 1 mol%, Atto 655D ppE, was used to eliminate the contributions form lateral diffusion to the observed fluorescence fluctuations. At such a high concentration, fluorescence intensity fluctuations arising from dye diffusion in and out of the confocal volume are negligible. Supplementary Fig. 2 and Supplementary Note 233. Furthermore, it's worth noting that no photobleaching was observed, attributed to the utilization of low excitation power for samples with high dye concentrations. The adhesion process was monitored using MIET imaging spectroscopy. Figure 1c shows the calculated dependence of lifetime and brightness as a function of height from the silica surface. See section Methods. This allowed us to translate a measured lifetime into an axial distance value using this lifetime distance calibration curve. In this study, only the membrane of the GUV was labeled, and its height over the surface was determined from the lifetime values of the labeling fluorophores. The fluorescence imaging and lifetime measurements for the samples were conducted using confocal scanning microscopy, employing pulsed laser excitation and time-correlated single-photon counting, TCSPC, for fluorescence lifetime measurements, as detailed in the Materials and Methods section. For calculating FLIM, we constructed TCSPC curves for each pixel and fitted these curves with a multi-exponential decay model, giving us a mean fluorescence lifetime for each pixel 34. Initially, we scanned the samples to identify suitable GUVs, including only those with a diameter exceeding 15 micrometers for further analysis. In Fig. 2a, one can observe a fluorescence FLIM image of GUVs containing ECAD on the SLBs after 1H of incubation. The FLIM image reveals 2 distinct binding states in the proximal membranes of the GUVs. The lifetimes and contact areas of the proximal membranes for GUV2 and GUV3 are significantly shorter and smaller than those of GUV1. The lifetime distribution for the contact area is uniform for GUV2 and GUV3, while it displays more heterogeneity for GUV1. Utilizing the MIET calibration curve depicted in Fig 1c, we transformed the FLIM images into height images. These height images revealed that the average bottom height of GUV2 and GUV3 above the surface is 35 plus or minus 1 nanometer, while GUV1 exhibits a greater height of 56 plus or minus 9 nanometers. It's important to note that the fluorescent lipid molecules are distributed in both leaflets of the GUV. Therefore, the calculated height represents the distance between the center of the lipid bilayer of the GUV and the surface of the MIET substrate. Factoring in the thickness of the SLBs and GUVs, approximately 4 nanometers, as well as the hydration layer, about 2 nanometers 31, we calculated that the membrane-membrane distance for GUV2 and GUV3 is approximately 27 nanometers, whereas for GUV1 it measures roughly 48 nanometers. We assumed that GUV2 and GUV3 formed the cis-cluster junctions, and calculated the EC5 distance by taking the size DFC of the FC fragment into account. DFC equals 7 nanometers, as determined from the crystal structure 35, as well as the tilt angle of the ECAD in the clustered state, approximately 30 degrees, determined from the crystal structure 2528. The estimated EC5 distance for GUV2 and GUV3 is around 20 nanometers, which closely aligns with the crystal structure of cis-cluster, 19 nanometers, suggesting that GUV2 and GUV3 have indeed formed the cis-cluster junctions. However, we could not precisely calculate the EC5 distance for GUV1 because unlike cis-cluster junctions which have a well-defined orientation on the membrane 25-28, ECAD monomers, or FC fragments, can be oriented at a range of angles on the membranes, or even lying flat on the membranes. Due to the observed inhomogeneity in GOV1, classifying the binding state for GOV1 solely based on its average height proves challenging. This observation implies that, in addition to the ECADs on the surface of the membranes, both GOV and SLB, that culminate in the final cis-clustered state, as seen in GOV2 and GOV3, some ECADs do not contribute to the formation of the ultimate cis-cluster. In fact, during our 2H observation period, membranes such as GOV1 consistently maintained this heterogeneous pattern. This observation aligns with a previous report indicating the existence of a nucleation process during ECAD junction formation, resulting in an all-or-nothing characteristic 23. In the initial phase of cis-cluster interface formation, we continuously monitored the ECAD-mediated adhesion process via FLIM imaging. See Supplementary Movie 1. Initially, the giant unilamellar vesicles GUVs positioned themselves above the SLBs to establish a contact zone. This contact zone subsequently contracted during the adhesion process due to the enrichment of cis clusters. To track the height variations throughout this process, we performed point measurements to record fluorescence intensity traces, supplementary Fig. 5. Subsequently, we transformed these fluorescence intensity traces into height traces by binning them at a 100 millisecond resolution. For each time bin, containing between 1 × 104 and 5 × 104 photons, we constructed a fluorescence decay histogram and determined the fluorescence lifetime using a multi-exponential decay model. The obtained lifetimes were then converted into height values utilizing the calibration curve presented in Fig. 1c. A typical height trace representing one GOV adhesion event is illustrated in Fig. 2d, which can be segmented into 3 distinct phases. The initial black region in Fig. 2d signifies the waiting period preceding ECAD clustering. The subsequent red segment corresponds to the clustering process itself, and the subsequent blue area represents the time trace of ECAD after clustering. It should be noted that the waiting times depicted in Fig. 2de do not precisely correspond to the initiation of adhesion. This discrepancy arises from the scanning of the sample before conducting the point measurement, leading to a delay in capturing the exact onset of adhesion. During the course of the curve depicted in Fig. 2d, the membrane initially exhibited significant height fluctuations within the range of 45 to 55 nanometers, black region. Subsequently, it gradually decreased to a lower height of approximately 31 nanometers, with reduced fluctuations, blue region. The initial height range of 45 to 55 nanometers corresponds to a minimum EC5 distance of 23 to 33 nanometers, assuming no tilt of the FC fragment. In contrast, the final height of approximately 31 nanometers corresponds to an EC5 distance of 16 nanometers. Assuming a tilt angle of 30 degrees for the ECAD at cis-cluster state, we attribute this to the conversion of ECAD from its dimeric state, SX dimer, to a final cis-clustered state. It is worth noting that we cannot directly observe monomer to dimer formation due to the fast dimerization rate, 3.8 times 104s minus 1/1620 of ECAD monomers. However, this does not preclude the existence of monomers in the initial state, because ECADs dynamically transition between different states. In Fig. 2d, the red segment highlights the phase of cis clustering. We discovered that this clustering strongly depends on the mobility of the membrane. For instance, when we replace the partially fluid SLB, composed of DPPC plus NBDPC, with a fluid SLB made of pure SOPC lipid, supplementary Fig. 4 and supplementary Note 3, the clustering rate increased by at least an order of magnitude. Fig. 2e, further detailed in supplementary Fig. 5 and supplementary Note 4. In instances where cluster formation did not occur, the lower membrane of a GOV exhibited continuous fluctuations at a consistent height, as visually demonstrated in supplementary Movie 2. In our MIET measurements, the GOV membrane fluctuates above the gold-coated surface, causing variations in fluorescence intensity, Fig. 1c. Importantly, we verified that these intensity fluctuations were the result of vertical displacements of the membrane and not caused by the lateral diffusion of individual fluorophores. This is due to the high fluorophore label concentration, 1 mol%, which excluded any contribution from the lateral diffusion of the fluorophores to the observed intensity fluctuations, supplementary Fig. 2 and supplementary Note 2. Furthermore, we conducted point measurements on this fluctuating membrane and analyzed the time trace of fluorescence intensity, which was constructed over time bins with a width of 10 milliseconds. Surprisingly, we discovered that the intensity distribution exhibited an asymmetry, and numerous dip signals were evident in the intensity time traces, Fig. 3a and supplementary Fig. 6. As a control, we did not observe such dip signals in a GOV membrane lacking ECAD or in a membrane after the formation of the cis cluster, Fig. 3b. Returning to the first case that led to the formation of the final cis cluster, we also observed these dip signals in certain time traces within the waiting time interval preceding ECAD clustering, Fig. 2e inset. To unravel the origin of the dip signals, we employed a two-threshold method to demarcate the dip signal, referred to as the low state, blue area, from the baseline, referred to as the high state, red area, within the intensity time trace, see supplementary Fig. 8 and supplementary Note 6. Subsequently, by accumulating decay times based on their photon counts for each time bin in the time trace, we constructed fluorescence lifetime decay histograms for these two distinct states and determined their respective fluorescence lifetimes, Fig. 3c. By fitting the fluorescence decay curves using a multi-exponential model, we extracted lifetimes of 1.65 nanoseconds for the high state and 1.51 nanoseconds for the low state, respectively. These lifetimes corresponded to heights of 55 nanometers and 47 nanometers above the silica surface. We analyzed 45 fluorescence time traces, each lasting for 5 minutes, from 27 GUVs. For the heights of the high state and low state, we obtained values of 52.8 plus or minus 4.9 nanometers and 48.8 plus or minus 5.6 nanometers, respectively, Fig. 3c. We attributed the high and low states to membrane positions associated with the ECAD S-dimer, or monomer, and X-dimer, respectively. This is because the brief duration of the low state in the intensity time trace corresponds to the short-lived and unstable structure of the X-dimer, and the X-dimer configuration should be observed at a lower height than the S-dimer, Fig. 1a. The low state should not be observed for the cis-clustered state, as it is a stable state. It has been reported that the rate constant of unbinding CISX and CISS to X-dimer and S-dimer is around 0.1 s minus 1, corresponding to a mean unbinding time of 100 s 1620. Furthermore, the dip signals in the intensity trace do not result from osmotic or thermally induced membrane fluctuation because these two effects would only lead to a symmetric height distribution, Fig. 3b. Next, we conducted experiments to validate that the consecutive dip signals emanate from the continuous formation of X-dimers within the confocal volume, rather than from the lateral diffusion of X-dimers. This confirms that X-dimers formed within the confocal volume and did not undergo significant diffusion in and out of the confocal volume. Supplementary Fig. 4 and supplementary Note 5. It's essential to highlight that during a point measurement with our confocal microscope, the observed signal represents a spatial average over the size of the excitation focus. Since numerous ECAD molecules are present within the confocal spot, and not all of them are involved in the conversion process, the measured height values of the membrane cannot be directly converted to the EC5 distances for a specific dimer state. However, MIET is capable of capturing any small height changes due to its high spatial resolution. It is essential to note that ECAD-mediated intermembrane adhesion is a multifaceted process with various pathways, including monomer-involved routes. These pathways encompass interactions such as monomer binding to S-X-dimers, backward unbinding from S-X-dimers to monomers, and cis-binding unbinding of monomers. Unfortunately, in our measurement, we cannot distinguish these specific monomer-involved processes. This limitation arises from the fact that our measurement relies on monitoring membrane height, and monomers exist in a free state without a distinct height signature. Consequently, changes in monomer populations do not induce discernible alterations in membrane height. For instance, consider the scenario of backward unbinding from an X-dimer to monomers. Given that a significant majority of ECADs are in an S-dimeric state, even if an X-dimer dissociates into monomers within each membrane, the membrane's height would still primarily reflect that of an S-dimer. Therefore, we cannot definitively conclude that the abrupt transition from the low state to to the high state in the time trace exclusively represents the process of converting X dimers to S dimers. This transition encompasses a combination of two potential pathways: X dimer to S dimer conversion and X dimer to monomer conversion. We further delved into the membrane fluctuations and displacements induced by the formation of X dimers. To achieve this, we computed membrane heights for a binning time of 100 milliseconds from the intensity trace, and conducted an analysis that considered both fluorescence intensity and membrane height fluctuations, as computed from the fluorescence lifetime, throughout the measurement. As illustrated in Fig. 3D, a two-dimensional 2D histogram reveals correlations between emission intensity and membrane height for the ECAD-modified membranes. This histogram depicts a positive correlation between fluorescence intensity and membrane height consistent with the theoretical intensity height curve presented in Fig. 1c. The height distribution spans a wide range, varying from 40 nanometers to 75 nanometers, with a predominant concentration around approximately 60 nanometers. In contrast, membranes lacking ECAD exhibit a narrower height range, spanning from 30 nanometers to 45 nanometers. Moreover, for membranes in the cis-clustered state, the height distribution is even more confined, ranging from 32 nanometers to 36 nanometers. Notably, the formation of X dimers leads to increased membrane fluctuations compared to the other two control membranes in the experiment, Fig. 3e-F. This increased fluctuation arises from a combination of two contributing factors. Firstly, strong thermal fluctuations occur as a result of osmolarity differences. Secondly, dynamic structural changes within dimers contribute significantly to this enhanced fluctuation. To quantitatively assess membrane fluctuation, we calculated the membrane displacement autocorrelation function, GHT, based on the fluorescence intensity autocorrelation function, GIT. As demonstrated earlier, fluctuations in fluorescence intensity exclusively originate from axial displacements of the membrane. Consequently, we can convert intensity correlation curve GIT into a height correlation curve GHT by utilizing the relationship between fluorescence intensity and membrane height. See materials and methods. This is done by using the relation GIT equals less than delta H, 0 delta HT greater than, where delta HT represents the instantaneous membrane height. The height correlation function provides the root mean squared displacement psi equals delta H2 of the membrane, as well as the relaxation time tau asterisk, defined as the time point at which GHT has declined to half of its maximum value 36. As illustrated in Fig. 3 grams, the displacement amplitude psi for a GOV membrane with ECAD is 6.9 plus or minus 1.5 nanometers, whereas it is psi equals 2.9 plus or minus 0.3 nanometers for the membrane lacking ECAD. In the latter case, the amplitude of membrane fluctuations is solely determined by the difference in osmolarity. Following the formation of the final cis clusters, membrane fluctuations become negligible and fall below the detection threshold, Fig. 4 grams. Furthermore, the relaxation time of the membrane fluctuations for GUVs with ECAD tau asterisk equals 0.1 s is notably slower than that for GUVs without ECAD tau asterisk equals 0.04 s. This difference is attributed to the slower structural interconversion between S and X dimers in the presence of ECAD. To dissect the formation dynamics of the ECAD X dimer, we individually annotated each intensity time trace by the time spent in the high state, H, and low state, L, between consecutive height switching cycles. These durations are denoted as the waiting time for each state, T, where I equals H or L as shown in Fig. 4a. These waiting times represent the durations required to transition to the next state. For calculating the average times, we first extracted all waiting times, both high and low states, from 25 individual intensity time traces, each lasting 600 s, and then calculated the mean waiting time across all waiting times, greater than 13,000 events. TH equals 0.41 plus or minus 1.29 s and TL equals 0.046 plus or minus 0.084 s. Here, denotes the averaging time, and errors are determined from the standard deviation of 13,160 switching cycles. The observed large error values in waiting times might be attributed to the fact that ECADs on each membrane are not solely monomers. X dimers formed from the cis dimers or cis oligomers dramatically differ in stability. Additionally, measurements using GUVs functionalized with a higher concentration of ECAD EC show a decrease in the average high-state waiting time to 0.25 plus or minus 0.56 s, while the low-state waiting time does not vary significantly, 0.051 plus or minus 0.098 s. Supplementary Fig. 6. This suggests that the measured low-state lifetime genuinely reflects the lifetime of the X-dimer. Using these averaged waiting times, we can calculate the transition rate constants for switching between the low and high states. KL right pointing arrow H equals 1, TL equals 22 plus or minus 39s, minus 1, and KL right pointing arrow H equals 1, TH equals 2.4 plus or minus 7.6 s minus 1. This results in an equilibrium constant, K equals KH right pointing arrow L per kiloliter right pointing arrow H, of approximately 0.11. Intriguingly, the rate constants for converting low state to high state is of the same order of magnitude as the rate constants for conformational switching between the S-dimer and X-dimer in solution, as measured by NMR relaxation dispersion spectroscopy. Kx right pointing arrow S equals 86 s minus 112. This further suggests that the transitions between the high and low states in the fluorescence time trace primarily arise from conformational changes between dimers. As an alternative method for determining the equilibrium constant for the transitions between high and low states, we employed a recently developed method called shrinking gate fluorescence correlation spectroscopy, SGFCS 37. In SGFCS, one generates a set of fluorescence intensity autocorrelation functions, IACFs, using distinct subsets of photons falling into different time windows on the nanoseconds timescale following laser pulses. If dynamic changes are occurring, the amplitudes of the IACFs should incrementally increase with a longer time window. As depicted in Fig. 4b, the IACFs for the membrane with ECAD clearly exhibit an increase in amplitudes as the time window becomes smaller, in step sizes of 0.5 nanoseconds. By fitting the curve amplitudes against window width, we determined the equilibrium constant to be 0.10 plus or minus 0.013. Fig. 4b, inset, and C-section methods for details. Interestingly, this equilibrium constant obtained using SGFCS closely matches that calculated from the waiting times. This agreement strengthens the conclusion that the dynamic transitions between high and low states primarily result from conformational changes between dimers. The distribution of individual waiting times follows an exponential decay function for both TH and TL, both displaying broad distributions, Fig. 4c. The typical trajectory in supplementary Fig. 9 illustrates the stochastic nature of waiting times for each state. This raises the question of whether these fluctuations in waiting times for each state exhibit temporal correlations. In other words, can the appearance of two consecutive waiting times be statistically correlated with temporal changes in the structures of the dimers? To investigate these potential correlations, we evaluated the 2D difference histogram, defined as delta Tn plus 1 equals Ptn plus 1 minus Ptn times Ptn plus 1. For the waiting times of each state from two adjacent cycles. Supplementary Note 7. This analysis involves calculating the joint probability for two waiting times, denoted as Ptn, Tn plus 1, supplementary Fig. 10, and comparing it to the probability of no correlation between these two times, which is represented by the product of the individual waiting time probabilities, Ptn, times Ptn plus 1. This statistical test has previously been employed in the analysis of dynamic disorder in enzymatic catalysis 383940. If there is no dynamic disorder, the joint probability P(Tn), Tn plus 1, and the product of individual probabilities Ptn times Ptn plus 1 would be the same. Supplementary Fig. 11. If they are different, it hints at the presence of a memory effect induced by conformational changes, where the system remembers its conformation for an extended period. We conducted an analysis of waiting times from all analyzed trajectories, n equals 25, each spanning a 600s experiment. Notably, we observed distinct waiting time distributions for the high and low states. As depicted in Fig. 4c-d, the waiting times for the high state did not exhibit significant time-correlated features. In contrast, for the low state, we identified a weak and broad diagonal feature in the waiting time distributions. This diagonal feature suggests that longer waiting times are less likely to be followed by shorter ones and vice versa. This intriguing pattern implies a form of temporal memory within the system. Remarkably, this correlation in waiting times for the low state persisted even when considering cycle lags of 20, corresponding to a time range on the order of seconds, supplementary Fig. 11. Such memory effects have been previously observed in enzymatic catalysis and attributed to conformational fluctuations in individual enzymes 38, 39, 40. However, in our experimental context, the low states result from the formation of numerous X-dimers, with ECAD molecular densities estimated to be approximately 100 to 700 molecules per square micrometer within each membrane 23. Consequently, the observed correlation or memory effect in the low state is less likely to stem from the conformational dynamics of individual X-dimers. Instead, it may arise from interdimer communication among the ECAD dimers. Specifically, the presence of X-dimeric states can influence the structure of nearby S-dimers by altering the membrane-membrane distance, potentially affecting the stability and persistence of X-dimers formed from these S-dimers. To further explore the role of membrane fluctuations in ECAD adhesion, we increase the osmolarity difference between the interior of the GUVs and the external buffer from 70 mosm/L to 170 mosm/L, supplementary Fig. 14. This increase led to a corresponding rise in membrane fluctuation amplitude, from 2.9 plus or minus 0.3 nanometers to 6.7 plus or minus 2.2 nanometers. Notably, under these enhanced membrane fluctuations, we observed no transitions between high and low states, and ECADs rapidly formed the final cis cluster state. This behavior may be attributed to the larger membrane fluctuations weakening the stability of both X and S dimers, thereby facilitating the formation of the stable cis cluster state. The observed dynamics of X-dimer formation and associated membrane displacement may represent early stages of cell-cell adhesion, potentially playing a role in cellular sensing or recognition processes. Discussion. We employed MET spectroscopy to probe membrane-membrane adhesion facilitated by ECADs on each membrane. Our investigations centered on a biomimetic system designed to emulate ECAD-mediated cell adhesion. Specifically, we explored adhesion events of the EC domains of ECADs, which were positioned between SLBs and GUVs, consistent with earlier reports 23 highlighting the all-or-nothing nature of ECAD junction formation. Our observations revealed two markedly distinct adhesion conformations. In the first scenario, we observed conventional ECAD-mediated adhesion, resulting in the formation of a cis cluster between the two membranes. Notably, we observed variations in membrane height during adhesion. Leveraging MET, we determined membrane height values at different stages of this process. These height values, or membrane-membrane distances, coupled with crystal structure information for ECAD and ECAD junctions, led us to conclude that ECAD experiences a transition from an initial dimeric state with a larger membrane-membrane distance to a final stable cis-clustered state with a smaller distance. In contrast, the second scenario involved the observation of only the dimeric state without further clustering. We detected continuous dip signals in the fluorescence intensity traces. Through analysis of the EC5 distance derived from these dip signals, our data strongly support the hypothesis of an X-dimer-dependent mechanism for wild-type ECAD-mediated membrane adhesion, with the dips indicative of X-dimer formation. Furthermore, we uncovered the kinetics of X-dimer formation and dissociation. The X-dimer exhibited a brief lifetime of approximately 50 milliseconds, underscoring its role as a short-lived intermediate state. Our MET-based analysis extended to quantifying membrane fluctuation amplitudes. We discovered that the conformational dynamics of dimers lead to more pronounced membrane displacements. Interestingly, these membrane displacements triggered a memory effect among X-dimer formations, hinting at mutual cooperativity between membrane displacement and transdimers. While prior simulations and experiments have underscored mutual cis-trans cooperativity in ECAD, stemming from apparent allosteric enhancement, our results suggest a mechanism whereby membrane displacement also contributes to this cooperativity. Furthermore, our method presents our approach to studying the stability and dynamics of X-dimers and S-dimers. In our biomimetic system, osmolarity difference and Ca2+ concentration emerge as the two most critical factors influencing E-cadherin adhesion dimers. For instance, under a larger osmolarity difference, 170 mosm/L, No GUVs exhibited high-state low-state dynamics, and all GUVs rapidly transitioned to the final cis cluster state, supplementary Fig. 14. Moreover, in the absence of Ca2+, no E-cadherin-mediated adhesion between GUV and SLB was observed, supplementary Fig. 15. A follow-up study employing our MIET method with systematic variations in osmolarity difference and Ca2+ concentration could provide deeper insights into their effects on the dynamics and stability of X-dimers and S-dimers. In summary, our utilization of MIET spectroscopy and imaging enabled us to monitor the height variations in a model ECAD-modified membrane system. We investigated the kinetic and dynamic processes underlying ECAD-mediated membrane adhesion, pinpointing various ECAD states based on membrane height, and studying their evolution during adhesion. Notably, we unraveled the existence of an X-dimeric intermediate state characterized by its transient nature and its significant impact on membrane displacement, which in turn influences the formation and stability of subsequent X-dimers. In particular, we found evidence that X-dimers communicate with one another through membrane displacement. These insights are crucial for understanding the role of ECAD junctions in maintaining cell-cell adhesion, tissue organization, and physiological processes. Further, our findings underscore the potential of MIET spectroscopy and imaging as a potent technique for elucidating subtle structural changes in membrane and membrane protein organization.